CNA Infection Control Study. Welcome to the quiz. Question 1. The autonomic nervous system has two major divisions. Which division is characterized by long preganglionic and short postganglionic neurons and uses acetylcholine as its primary neurotransmitter at both ganglia and effector organs? Option A. The somatic motor system. It innervates skeletal muscle. Option B. The sympathetic division. It uses norepinephrine at most effectors. Option C. The enteric nervous system. It is intrinsic to the gut. Option D. The parasympathetic division. It is rest and digest. The answer is D. The autonomic nervous system ANS controls involuntary functions like heart rate and digestion. It has two main branches with opposite effects. The parasympathetic division is responsible for rest and digest activities, conserving energy. Its anatomical hallmark is long preganglionic neurons that originate in the brainstem or sacral spinal cord and travel a long distance to ganglia located very near or within the target organs. Because the ganglia are close, the postganglionic neurons are very short. Both the preganglionic neuron at the ganglion and the postganglionic neuron at the effector organ release the neurotransmitter acetylcholine ACH. This is in contrast to the sympathetic division. Option A is incorrect. The somatic motor system controls voluntary skeletal muscles and uses a single neuron, not a two-neuron chain that releases ACH at the neuromuscular junction. Option B is incorrect. The sympathetic division has short preganglionic and long postganglionic neurons and uses ACH at the ganglion, but norepinephrine at most effector organs except sweat glands. Option C, the enteric nervous system, is a complex network within the GI tract wall and is considered a separate division of the ANS, but it is not characterized by the long pre/short postganglionic structure of the parasympathetic outflow. Question 2. Hearing relies on the transduction of sound waves into neural signals. In the organ of Corti, what is the specific mechanism by which stereocilia bending leads to depolarization of the hair cell? Option A. The bending opens voltage-gated sodium channels. Option B. The bending opens mechanically gated potassium channels, allowing K influx from the endolymph. Option C. The bending closes chloride channels, leading to a more negative potential. Option D. The bending triggers the release of glutamate from the base of the cell. The answer is B. Sound waves cause vibrations in the fluid-filled cochlea, which moves the basilar membrane. Sitting on this membrane is the organ of Corti, which contains the sensory hair cells. Each hair cell has a bundle of stiff hairs called stereocilia on top. The tips of shorter stereocilia are connected to the sides of taller ones by tiny filamentous tip links. When sound vibrations move the basilar membrane up and down, the stereocilia bundle bends. Bending in the direction of the tallest stereocilia stretches the tip links. This stretching pulls open mechanically gated ion channels at the tips of the stereocilia. The unique feature here is that the fluid surrounding the stereocilia endolymph is unusually rich in potassium K. Therefore, when these channels open, it is primarily potassium ions K that rush into the hair cell down their electrochemical gradient. This influx of positive ions depolarizes the hair cell, makes it less negative inside. This depolarization then opens voltage-gated calcium channels at the base of the cell, leading to neurotransmitter glutamate release. Option A is incorrect because the initial channels opened are mechanically gated, not voltage-gated. Option C is incorrect. Depolarization is caused by an influx of positive ions, not by closing channels. Option D describes a consequence of depolarization neurotransmitter release, not the initial transduction mechanism. Question 3. Neurotransmitter action must be terminated. At the cholinergic synapse, what is the enzyme that rapidly hydrolyzes acetylcholine into inactive metabolites, and where is it located? Option A. Catechol-O-methyltransferase COMT, located in the synaptic cleft. Option B. Monoamine oxidase MO, located in the presynaptic mitochondrial membrane. Option C. Acetylcholinesterase ACHE, located on the postsynaptic membrane. Option D, glutamate decarboxylase, located in the cytoplasm of inhibitory neurons. The answer is C. For a synapse to function properly, the signal must be turned off quickly after it is sent. At synapses that use acetylcholine ACH as a neurotransmitter, cholinergic synapses, like the neuromuscular junction, the primary mechanism for terminating the signal is enzymatic breakdown. The specific enzyme is acetylcholinesterase, ACHE. ACHE is anchored into the postsynaptic membrane, with its active site facing the synaptic cleft. When ACH is released and binds to its receptors, ACHE is right there to break it down. It hydrolyzes ACH very rapidly in milliseconds into two inactive products, choline and acetate. This rapid breakdown ensures that ACH does not continue to stimulate the postsynaptic cell, allowing for precise control of muscle contraction or nerve signals. Option A, COMT, breaks down catecholamines like dopamine and norepinephrine. Option B, MAO, also breaks down catecholamines and serotonin inside neurons. Option D, glutamate decarboxylase, is the enzyme that makes the inhibitory neurotransmitter GABA from glutamate. Question 4. Gas exchange in the alveoli follows Fick's law of diffusion. Which factor, when decreased due to conditions like pulmonary fibrosis or ARDS, most directly impairs the diffusion of oxygen into the pulmonary capillaries? Option A, the partial pressure gradient of O between alveoli and blood. Option B, the thickness of the alveolar capillary membrane diffusion distance. Option C, the solubility coefficient of oxygen in plasma. Option D, the cardiac output and pulmonary blood flow. The answer is B. Fick's law states that the rate of diffusion of a gas across a membrane is proportional to the surface area, the diffusion coefficient related to solubility and molecular weight, and the partial pressure gradient, and inversely proportional to the membrane thickness. In diseases like pulmonary fibrosis or acute respiratory distress syndrome ARDS, the lung tissue becomes scarred, inflamed, and thickened. The primary pathological change that directly hinders oxygen diffusion is an increase in the thickness of the alveolar capillary membrane. This is the barrier oxygen must cross from the alveolar air space through the alveolar epithelium, basement membrane, and capillary endothelium into the blood. When this barrier is thickened, increased diffusion distance, oxygen molecules take longer to travel across it, even if the pressure gradient is normal. This can lead to hypoxemia, low blood oxygen that is not easily corrected by simply giving more oxygen. Option A, the pressure gradient, is usually normal or even increased in these diseases if the patient is given supplemental oxygen, yet hypoxemia persists. Option C, the solubility coefficient, is a constant physical property of oxygen and does not change in disease. Option D, cardiac output, affects overall oxygen delivery but not the fundamental diffusion process at the alveolar level. Question 5. The majority of carbon dioxide CO is transported in the blood in what specific form, and where does the conversion into this form primarily occur to facilitate this transport? Option A, dissolved as CO gas, occurs in the systemic capillaries. Option B, bound to the heme group of hemoglobin, occurs in the plasma. Option C, as bicarbonate ions HCO, occurs within red blood cells via carbonic anhydrase. Option D, as carbaminohemoglobin, occurs in the alveoli during exhalation. The answer is C. Carbon dioxide produced by tissues is carried in the blood back to the lungs. The vast majority, about 70%, is transported in the form of bicarbonate ion HCO. This conversion is crucial because it allows the blood to carry a large amount of CO without causing a massive increase in partial pressure. The process starts when CO diffuses from tissues into the blood and then into red blood cells RBCs. Inside RBCs, an enzyme called carbonic anhydrase rapidly catalyzes the reaction of CO with water HO to form carbonic acid HCO. Carbonic acid immediately dissociates into a hydrogen ion H and a bicarbonate ion HCO. The bicarbonate ion is then exchanged for a chloride ion Cl out of the RBC via the chloride shift band 3 protein, allowing HCO to travel dissolved in the plasma. This efficient system allows for the bulk transport of CO. Option A is incorrect. Only a small fraction, about 7 to 10 of CO, is transported dissolved directly in plasma. Option B is incorrect. CO does not bind to the heme group of hemoglobin, that's where O binds. Some CO, about 20, binds to the amino groups of hemoglobin-globin chains, forming carbaminohemoglobin, but this is not the primary form. Option D is incorrect. The formation of carbaminohemoglobin occurs in tissues, not primarily in the alveoli. Question 6. Surfactant, produced by type 2 pneumocytes, is crucial for normal lung function. What is its primary physiological role at the alveolar air-liquid interface? Option A. To facilitate the diffusion of oxygen across the membrane. Option B. To provide an immunological defense against inhaled pathogens. Option C. To reduce surface tension, preventing alveolar collapse atelectasis at end expiration. Option D. To increase lung compliance by thickening the alveolar walls. The answer is C. The inside of the alveoli is lined with a thin layer of fluid. At any air-liquid interface, water molecules are strongly attracted to each other, creating a force called surface tension. This surface tension acts like an elastic film that constantly tries to shrink the alveolus, making it hard to inflate and tending to collapse it, especially at the end of expiration when the alveolus is smallest. Pulmonary surfactant is a complex mixture of lipids and proteins secreted by type 2 alveolar cells. Its primary and most critical function is to reduce this surface tension. The surfactant molecules, particularly dipalmitoyl phosphatidylcholine DPPC, insert themselves between water molecules at the surface, breaking their strong cohesive forces. By lowering surface tension, surfactant, 1. Dramatically decreases the work of breathing, increases lung compliance, 2. Stabilizes alveoli of different sizes, and 3. Most importantly, prevents alveolar collapse atelectasis at low lung volumes. Without surfactant, as in premature infants with respiratory distress syndrome, the lungs are very stiff and alveoli collapse with each breath. Option A. Surfactant does not directly facilitate gas diffusion. That depends on the thinness of the membrane. Option B. While some surfactant proteins have immune functions, this is not its primary role. Option D. Surfactant increases compliance by reducing surface tension, not by physically thickening walls. Great job! Don't forget to subscribe. Bye!